Hello friends, today we'll discuss about one of the topic of cell biology, that is cytoskeleton and cell mortality. ठीक है, तो cytoskeleton में basically आपका क्या क्या आता है? We have three different cytoskeletal components. One is microtubules, microfilaments and intermediate filaments. So first of all we'll discuss about microtubules. तो अगर हम देखें जो माइक्रोट्यूब्यूल्स हैं उसका जो डायमीटर होता है दैट इज 20 to 30 नैनोमीटर एंड योर सीलिया फ्लैजिला जिसका रोल मोटिलिटी में होता है, सो दे आर मेड अप ऑफ योर माइक्रोट्यूब्यूल्स एंड बिसाइड दैट वी हैव सेंट्रियोल्स. सेंट्रियोल्स जो होते हैं जिससे आपके स्पिंडल फाइबर्स ओरिजिनेट होते हैं और जिनके साथ आपके क्रोमोसोम्स अटैच करते हैं. ठीक है, तो अभी हम देखेंगे इनके बारे में कि सीलिया फ्लैजिला एंड सेंट्रियोल्स. ठीक है, सो बेसिक वी हैव ट्यूबलिन. ट्यूबलिन इज द प्रोटीन जिससे आपके माइक्रोट्यूब्यूल्स बने होते हैं. सो वी हैव टू डिफरेंट टाइप ऑफ ट्यूबलिन, दैट इज ट्यूबलिन ए एंड बी, एंड दीज ट्यूबलिन आर इन द फॉर्म ऑफ हेट्रोडाइमर. देयर इज वन ए एंड वन बी. ठीक है, सो वन ऑफ द क्वेश्चन दे आस्क इन एग्जाम्स, वी हैव सम ड्रग्स व्हिच इनहिबिट द assembly or disassembly of microtubules. So like we have few drugs like colchicine, vinblastine and vincristin. Colchicine, it is obtained from a plant that is Colchicum autumnale, and vinblastine and vincristin is obtained from Catharanthus roseus. ठीक है, so these drugs basically what they do, they inhibit assembly, because during cell division both assembly and disassembly of microtubules are important. So those drugs which either inhibit assembly or disassembly, they are interfering with the mitosis. So these three drugs, colchicine, vinblastine and vincristin, so what they do, they inhibit assembly of microtubules. And one we have another drug that is Taxol. Taxol is basically obtained from a plant that is yew tree, and we have the scientific name is Taxus baccata or Taxus brevifolia. So this particular drug inhibits disassembly of microtubules. ठीक है, so if we have a look at the arrangement of this tubulin in cilia and flagella, so there is 9+2 arrangement. ठीक है, so that we can have in diagram. So 9 is peripheral, we have nine doublets at periphery, and 2 is indicating that we have 2 central microtubules. So this is 9+2 arrangement. ठीक है, so 9 are peripheral, these are 9 peripheral doublets, and 2 we have central microtubules. So how come these doublets are linked? So this is one of the question they ask in exam. These doublets are linked through Nexin links. So there is a link between doublets, we call it Nexin link, and there is basically we have a subfiber A. So this subfiber A have a dynein arm. So this subfiber A, because we have two subunits A and B, A subunit have a dynein arm which is a high molecular weight ATPase. ठीक है, क्योंकि अभी हम पढ़ेंगे क्योंकि डाइनिन का रोल होता है मोटिलिटी के अंदर, क्योंकि अगर डाइनिन के पास एटीपेज एक्टिविटी नहीं है, सो देन देयर विल बी नो मोटिलिटी ऑफ द सीलिया और फ्लैजिला. ठीक है, सो देयर आर सम रेडियल स्पोक्स व्हिच आर कमिंग फ्रॉम द डबलेट्स. सो दीज आर योर रेडियल स्पोक्स. बिसाइड दैट वी हैव 2 central microtubules, so 9 peripheral and 2 central, and we have a sheath also. So this particular sheath is known as central sheath. Central sheath. And we have a term that is axoneme. So this 9+2 arrangement that we have of cilia and flagella, we have a term for this that is known as axoneme. Axoneme is the term applied to the axial basic microtubular structure of cilia and flagella, and it is the essential motile element. ठीक है, so beside that we have centrioles and basal bodies. ठीक है, because the cilia and flagella, so they are originate from basal bodies. Cilia and flagella originate from basal bodies. Originate from basal bodies. So if we have a look at the basal bodies and centrioles, so how they are different from cilia and flagella? So they have triplet. Like we have discussed, there are 9 doublets, but basal bodies and centrioles contain microtubular triplets. Basically they have 3 subunits A, B and C, and the arrangement we have in basal bodies and centrioles is 9+0. ठीक है, basically there are no central microtubules. In 9+2 we have your 2 central microtubules, but in the case of basal bodies and centriole the arrangement is 9+0, and because of 9+0 there are no central microtubules. So as we know, these cilia and flagella, they are responsible for motility, because the presence of cilia or flagella makes a cell motile. So what is the basis of this motility? So as these doublets are there, so these doublets, they slide over each other. When they slide over each other, then there is motility, then there will be movement of the cell. So the sliding of microtubular doublets, which is the basis of ciliary motion, is apparently due to a mechanochemical cycle involving ATPase present in the dyninam. ठीक है, क्योंकि जो हमने अभी पढ़ा कि सब फाइबर ए के अंदर एक डायनीनाम है, डायनीनाम के अंदर ATPase एक्टिविटी है, तो उससे जो एनर्जी आती है, दैट एनर्जी इज रिक्वायर्ड फॉर द स्लाइडिंग ऑफ डबलेट्स. ठीक है, सो इफ समहाउ दिस डायनीनाम इज नॉट फंक्शनल, so there is one of the syndrome that is immotile cilia syndrome. So in these patients, cilia and sperm are non-motile because there is lack or deficiency of dyninam. ठीक है, because dyninam is very important. If the dyninam is not functional or its ATPase activity is not functional, then it will lead to immotile cilia syndrome. Thank you.